All right, so I'm right outside the biology building. This is the back side of the rec center. And you can see in this image we have two different trees. We have a blue spruce on the right, which is a coniferous tree, and the leaves are needles. And then to the left we have a deciduous tree then with broad leaves. So for this lab I want you to pay attention to the following observation. So here's both of these trees. We have the deciduous tree on the left, and it has kind of the normal green color for the leaves. However, if we look at the blue spruce on the right, you can see that there is a blue tint to the leaves. So the question is, evolutionarily then, why has the blue spruce made these needles that are blue in color rather than the traditional green that you pretty much see on every single other tree around? Okay, so here's a closer view of the needles. So we have our blue spruce right here. You can see that it's coniferous with needles rather than leaves. And we can see up here at the top, this is in bright sunlight, sun is shining down. These needles are nice and blue, but if we look down here below, all of these needles below are green. If we look at this one right here, we can see that the tip of this one is blue, but further up the stalk it is green. If we look at this one though, the tip is not blue, it's green, and the whole thing is green. So the question is now, why would these be blue whereas these are green? And one potential hypothesis as to why these would be blue whereas these are green, if you notice, the blue ones then are in the bright sunlight hitting it. Our green ones then are in the shadier environments. So this could be an adaptation, and the blue part even possibly an adaptation for sun. In fact, we would probably think that it was an adaptation for sun, because the bright blue light then is hit directly by the sun. Whereas down here, these are going to be more hit by the shade. So for some reason then, as a hypothesis, we would think that the blue in these needles is an adaptation for improved photosynthesis in bright sunlight. Whereas down here, these are green because they're adapted to have better photosynthesis in a shady environment. Okay, so a big question would be, why then would blue needles in sunlight allow the blue spruce to photosynthesize better? To give you a little background information then, what I want to do is go in and look up what's called an absorption spectrum of photosynthesis. So I went into Google and I Googled an absorption spectrum of photosynthesis, and there's a lot of different good images. Let me choose one that I want. So right here is a good one. We'll click on that one, select it, and we'll go in here, and then I'll just go down and click on that image. So this is a pretty good image of what we're looking for. Here we have an absorption spectrum. An absorption spectrum then basically is looking at the absorption of the various pigments associated with photosynthesis. We have three pigments: chlorophyll A, chlorophyll B, and carotenoids. The y-axis you see is percent absorption. The x-axis is wavelength. The higher this absorption, then the more light is captured by the pigment, thus the more it has energy inside the pigment for photosynthesis. Okay. Now you'll notice that here we have visible light. So it's kind of interesting that these pigments are colored because they're absorbing in the visible spectrum that we see with our eyes. We have less energy over here at 700 nanometers. We have more energy here at 400 nanometers. If we go further this direction, we would even have more energy in ultraviolet light. But for some reason, our pigments are not absorbing in this ultraviolet light, and photosynthesis actually works within these wavelengths of light that are visible to our eyes, which is very interesting. And we'll actually talk about why that is later. Okay. Now what I want you to notice then is that we have these pigments. Chlorophyll A is a primary pigment. It absorbs here in green or in blue light. It has another peak absorbance here in red light, but it doesn't absorb anything here in this green, yellow, and orange region. It doesn't absorb down here, and it doesn't absorb up here. Chlorophyll B then has an absorbance peak right here in blue light. It has an absorbance peak here in green light. You can see that if we combine chlorophyll A and chlorophyll B, we have a bigger absorbance here in the red and a bigger absorbance here in the blue.
Carotenoids then is a third group of pigments, and they absorb here mainly, we have a big peak over here in this green range. So if we combine all three of these pigments together, we can see we can absorb and gather light in a bigger range here and a bigger range here. However, carotenoids have also been shown that there are different types of carotenoids. We have keratines and xanthophylls. Xanthophylls is a type of carotenoid that absorbs here, but it actually acts as what's called a photodissipation agent. Rather than absorbing light and sending it to photosynthesis, it absorbs the light in this blue range, all here, and basically dissipates that light. In other words, gets rid of it and doesn't send it to photosynthesis at all. In other words, in essence, then, xanthophylls are stealing light in this blue range and not sending it to photosynthesis. And it turns out that they're acting as a sort of a sunscreen, because one of the problems with photosynthesis is if you're in too much light and you have too much energy being absorbed, oxygen radicals are produced, and those oxygen radicals are highly reactive and can damage the plant. Basically, they're oxidants. So like you eat antioxidants to get rid of those oxidants. In plants, if you have too much light, then oxidants are formed. Now over here, then we can see we have big absorption in blue, and our xanthophylls are stealing it in blue light. So if we look at plants that are typically grown in high light condition, those plants have a lot of xanthophylls, taking away this energy in the blue light. Energetically, though, then if you have to manufacture this pigment all the time, it breaks down, it builds up, you manufacture it, that can expend a lot of energy. If we think about the blue spruce in highlight, the blue spruce might be doing the same thing that these xanthophylls are doing, simply by changing that cuticle and reflecting off the blue light for that photoprotection.
Now, to test our hypothesis, what we're going to do is I'm going to take a sprig. I'll basically go in here and cut off a sprig of blue. We'll take it back to the lab and we will run our photosynthesis trials, looking at blue light and red light and measuring the change in CO2 for when blue light is shown onto the tree versus red light. Then I'm going to go back and we'll cut off one of these, which is green in color. And then we'll do a blue light treatment and a red light treatment again, measure photosynthesis under blue light and red light, and then compare the green form to the blue form in blue light versus red light.